Buenas tardes a todo el mundo, otra vez más en un nuevo DealÜP de Capital Cell, esta vez con el equipo de Flomics, que nos presentarán su oportunidad de inversión. Haré una presentación muy rápida de quién tenemos hoy con nosotros. Hoy tenemos a João y André, ambos cofundadores de Flomics, João que es CEO y André que es COO, que en castellano se confunde un poco. Y también nos acompaña con nosotros Lorenzo de Decelera Ventures, que nos dará la visión que siempre nos gusta dar del inversor de referencia, que está invirtiendo en esta ronda y en anteriores, si no me corrige después.
Encantado.
La idea del webinar la haremos totalmente en castellano. No sé si ya se ha colado alguien que no entienda el castellano. Si es así, que nos lo diga, porque le podremos ir a hacer traducciones. Sí que Joao va a
En
presentar en
inglés.
inglés. Sí, sí, André y Joao van a presentar en inglés, pero nosotros vamos a estar haciendo en castellano. Entonces, si hay alguien en inglés, pues que haga preguntas en inglés. Vamos a hacer lo que vosotros queráis. Como antes de que empecéis, Joao y André, ya voy diciendo que durante la presentación podéis levantar la mano o escribir las preguntas en el Q&A y las iremos leyendo. Siempre al final damos la presentación de primero de Joao y André, después dejaremos que hable Lorenzo, daremos un poco la visión de por qué Capital Cell invierte muy rápido y volveremos con el Q&A con vuestras preguntas. Así que con esto, Joao, André, presentaros y adelante.
Vale, pues empiezo yo, después paso la palabra a João y sigue con la presentación. Pues yo soy André Guedes, soy COO y también cofundador juntamente con João de este proyecto de Flomics. Mi background es física, yo no soy del mundo de biología, tengo un doctorado en física y ya estoy en el mundo de las startups hace muchos años. Flomics ya es mi quinta startup. Ya tuve un exit, ¿no? Trabajé muchos años en San Francisco, entonces tengo una experiencia muy interesante en el mundo de las startups, innovación, tecnología. Y aquí ahora en este proyecto, juntamente con João, algo muy relevante como detección precoz de cáncer, queremos tirar adelante y realmente hacer una diferencia en el mundo de la salud. ¿Vale? Bueno, y ahora paso la palabra a João, que se presente y sigue con nuestro pitch.
Muchas gracias, pues yo soy João, soy portugués también como André y mi background sí que es en biología o en biología computacional para ser más específico. Llegué a Barcelona para hacer el doctorado en el estudio de RNA y ahora en Flomics estoy un poco haciendo el seguimiento del conocimiento que he adquirido durante este periodo, intentando aplicarlo un poco más a la vida real, a la práctica clínica. Y Flomics sí que es mi primera startup, pero ya he participado en la fundación de algunas redes de emprendedores y de científicos con interés en aprender un poco más sobre el mundo. Y fue ahí que también que me formé un poco como emprendedor y donde maduré un poco la idea de transformar mi know-how en una empresa. Voy a compartir mi pantalla y os cuento la historia de Flomics. A ver si funciona. Asumo que todo está bien, nadie se está quejando.
Perfecto.
Entonces voy a empezar y como decimos anteriormente, lo voy a hacer en inglés para que todo el mundo pueda entender, porque es un idioma más científico internacional, digamos. Okay, so good afternoon. I am João, and this is the story of Flomics Biotech. And I'm sorry, but I always have to start with some bad news. 1/3 of the population is expected to be diagnosed with cancer at some point in their lifetime. And the worst part is that we are detecting this cancer when it's already too late, because mostly our cancers at the moment are being diagnosed when there's a trigger caused by the symptoms, which means that 60% of the cancers are detected in advanced stage, which increases by 300% the mortality, but also the costs associated with the treatment by the healthcare systems. Currently in the market, there are only 5 types of cancer that have early detection screening solutions, and this depends on the country or even the location that you live in, because not all hospitals, not all regions have access to the same technology. What is common is that most of these tests have a very low accuracy, so they are not very sensitive or not very specific, and it's very inconvenient for the patient because each test is only applicable for one type of cancer. So if you want to get these 5 types of cancer screened, you need to do 5 tests separately every year or every couple of years. That's why at Flomics, we are developing a blood test for the early diagnosis of cancer. So it's targeting at-risk individuals, so people that do not have any symptom yet, but have some risk factors such as age or the history of smoking or cancer in the family. They want to make sure that everything is fine before it's too late. They go to the doctor, they get a Flomics test, and by surveying the molecular signs in our blood, we can detect warning signs even before the first symptoms appear. In case we detect something that is associated with cancer, we warn the potential patient that will go into a confirmation standard, gold standard test, like a colonoscopy or a CT scan to the lung, whatever, which maximizes the chances of early diagnosis and opens the doors to better therapeutic options. Liquid biopsy is what we're talking about. So is that new? Maybe some of you already heard about liquid biopsies before. Mostly these liquid biopsies that are being developed, because no liquid biopsy for the early detection of cancer is in the market at the moment. Most of these tests are focusing in detecting circulating tumor DNA or circulating tumor cells, which have a lot of potential, but also a lot of drawbacks that I will tell you in the next slides. First of all, looking at circulating tumor DNA, which was the biggest promise in the field, maybe already 10 years ago, when people realized that it was possible to detect tumor DNA in our blood. So DNA that comes from the tumors in circulation in our blood. But more recently, some clinical trials show that it's not so widely applicable, so it's not effective for all types of patients. And some companies that raised a lot of money, like on the right, we have the example of Renome. They raised in the beginning of the year 250 million. They released very underwhelming results with performance of around 50% in some cancer stages. So not really useful or not really good enough to be a test implemented globally after such a big hype that was created around the technology of detecting DNA. That is why at Flomics, we are doing it differently. We are unleashing the power of cell-free RNA. So RNA is a molecule that our cells produce every minute in a different way, depending on the external stimuli that they are fighting. So we can say that since the blood is in contact with all our organs, the pool of RNAs that are inside our blood is like a snapshot of the current health status of an individual, because all the organs are releasing RNA into the blood. And we are looking at this RNA, or to be more precise, we are sequencing and quantifying this RNA by the usage of next generation sequencing and advanced bioinformatics with algorithms of machine learning that detect patterns associated with the disease. The field of RNA is quite young in scientific research, but it's growing super fast. So on the left, you have the number of times that the cell-free RNA is mentioned in scientific publications, and you can see that it boomed in the last 5-7 years. The peak was during the COVID. As you might be aware, the vaccines were generated by using of messenger RNA, which is one of the RNA types that exist. So everybody started looking at RNA, and after the hype of DNA, the hype of RNA came. And a lot of super interesting papers came out. We have here some examples where people start showing it was possible to detect signs of the disease, looking at RNAs in blood and other types of fluids or solid tissues. But as in any very new, novel technology, there are also some drawbacks. So it was found that in most of these papers, there were some problems in the studies. There was some contamination of DNA, contamination of bacteria, degradation of the sample, or even errors in the analysis that were done that were giving results that were not reproducible. So they were detected in a special set of samples that the scientists look at, but when tried to reproduce in different types of people, of patients, the results didn't hold. And that's why at Flomics, we decided to put all our energy in the development of a proprietary protocol for cell-free RNA sequencing, optimize and adapt to the challenges of using RNA in plasma. RNA is a very unstable molecule. That's why it causes so much problems, and that's why most of the people ignored it until 5 years ago, because it was thought that it was not possible to use it when it's outside of the cells in circulation, but it turns out it's possible. It's just a bit more complicated. And as I said, at Flomics, we developed this protocol that allows to sequence, quantify, read these molecules with at the maximum quality in the market. If we look at the plot in the right, we have here represented a quality of the RNA obtained, let's say. And in blue, you have the quality of Flomics. In 2019, actually, our quality was very low. We didn't know it was possible to do better at the time, until we start seeing some papers in the literature that have much better quality. And that's when we decided to start improving our quality. And progressively, we have been improving until the moment we are now, that we have the highest quality, so the highest value in the y-axis, but also the lowest variability. So most of our samples are in super high quality, while in other competitors, let's say, the quality is very variable, with some samples with very good quality, some samples with very low quality, which we don't want if you want to implement this in the clinical practice. In order to make sure that the results were really robust, what we did was to take different samples from the same person and consider replicates and sequence with our protocol in multiple times. And we expect that if we repeat analysis of the same person in a row, so in different days, but the same person, the sample should look like the same in theory. Unless it's affected by external factors that we cannot control. And what we did here is the 3 different patients represented with the 3 different colors. And what we see is that independently of the day that the sample is taken, the same patient, so the same color, always clusters together. So the samples group together by the number, by the patient that they come from, and not by the day, not by the hour, not by some random things that we cannot predict. So we have here one of the patients, here another patient, and here another patient, which tells us that the results are very reproducible and gives us good hopes to being able to replicate results in external cohorts. So now that we have this very good optimized cell-free RNA protocol, we want to demonstrate its usage in the clinical practice. And we are doing it by applying our know-how, our knowledge into the detection of cancer. So we are collaborating with different hospitals, including the Hospital Clinic from Barcelona, the Biobank from Andalusia, hospital from Montpellier, and other collaborators that are not here because we are always adding new people in our studies. But together we collected over 1,300 samples of colon, lung, breast, prostate, and pancreas cancer. What we are doing at the moment, this is still ongoing, is to quantify the different RNAs that exist in the samples of these patients, treat them with our bioinformatics pipeline, meaning we treat the data, we simplify, we correct for confounding factors, we reduce the noise, and we make sure that the data is with the highest quality possible in order to feed to a machine learning algorithm that we use to learn what are the differences between a colon cancer patient, a lung cancer patient, a breast cancer patient, and a healthy individual. So our goal is that in the end, we feed a sample that we don't know if they have one type of cancer, and the model will see this pattern is associated with colon cancer, or this pattern is associated with healthy individual, or this pattern is associated with pancreas cancer, all together. So all the different types of cancer will come out from one single sample at the same time. We've not needed to repeat tests, and just with one blood sample, we can survey 5 types of cancer now and even more in the future. But as I said, at the moment, we are still training this algorithm, meaning that we are still trying to improve our results. Nothing is final yet, but I can show you where we are. So at the moment, we are achieving a sensitivity of around 95% and a specificity of around 87%. And if you look here on the right, this is a two-dimensional representation of the last samples that we analyzed in-house in our laboratory. What we expect is that the healthy individual here represented in green and the cancer individual represented in red. And I'm sorry for the color-blind people. We didn't think about it. And this is super new, so it's not as beautiful as it should be. This is super fresh, but we expect the samples, if everything goes as planned, to separate by the cancer stage. And we can see that most of the green accumulate here, most of the red accumulate here, but there are some samples that are not correctly placed. That's why we don't have a 100% sensitivity or 100% specificity. But this is something that, as I said, we keep improving. So maybe in 1 month from now, as we keep accumulating more samples and the model keeps learning subtle differences between the samples, we will improve, and we hope that all the samples in the limit, all the samples will be separated by cancer stage, and we'll have the super high performance. We are doing this by looking at cell-free RNA. So we check the abundance of the different RNA molecules in our blood, in our plasma. But we are also exploring different ways of looking into data, because we know that more people are starting to look at RNA. So we need to find a differentiation. But what we are doing is that, so we're generating the data with our KICAS cell-free RNA protocol. Now we are finding different ways of generating more data points to be using machine learning. One of the most classical ways, and the one that we used until now, is the gene abundance, so the quantification, how many molecules of each RNA. But we are also exploring the possibility of using circular RNAs, which are a special type of RNA that are produced in our body and that have a special role in cancer. It's described in the literature. They are a lot more stable, these RNAs, because they have this circular form, which is much better anti-degradation enzymes. They are more difficult to find, more difficult to quantify. We are finding ways to quantify them. We are also looking at RNA that we have in our body that actually come from bacteria or virus. It is known in some cancers that our metagenome, the different set of bacteria we have, might influence our health status. What we're doing here is that from the same cell-free RNA experiment, we are quantifying human RNAs, but we're also quantifying bacterial RNAs, viral RNAs, and all of them will be fed into the machine learning algorithm. Also tissue deconvolution. So we calculate what is the contribution of each organ for the RNAs that we find in plasma. So as an example, a very simple example, we can consider that a healthy individual have 20% RNAs from liver, 20% RNAs from kidney, 20% RNAs from heart, whatever. If we find too many RNAs from liver, maybe it's a warning sign that something is wrong in the liver. So what we're doing is to give all this data, which together generates more than 100,000 data points of potential biomarkers. We feed all this data to our machine learning algorithm, and the algorithm is the one that will decide which information is helpful, which information can be used for the cancer detection, and uses it to make the classification. When we get to the market, we are envisioning a business model that partners with central laboratories. So at Flomics, we'll produce the cancer detection kit and also all these machine learning models that separate or that classify the samples. We do it in-house. We build simplify a box with all the reagents needed. Then we partner with central laboratories such as Unilab, Synlab, Etchevarne, etc. We capacitate them to perform the analysis in-house, so they will be the ones that will be in touch with the hospital, with the clinics, getting samples, taking care of the sample processing, distributing, etc. They will perform the cell-free RNA experiment, and then the interpretation of the data will be done in Flomics cloud. So we already built a cloud-based platform that we call Stratus that has our algorithms there, and then all the central labs will be able to get the data and upload it to our website and get the interpretation. Because the data that you generate in a cell-free RNA experiment, these over 100,000 potential biomarkers without the right interpretation, it's completely meaningless. It's too much information. It's impossible to understand what's there. Only a trained algorithm can understand and can make a simplification that can be translated into a medical report, let's say. We are aiming to enter the market in 2026 as a research use only, so before we get the certification, where hospitals can use it as a research tool, and then in 2027 as an in vitro diagnostic tool certified by the CE. And that's what we're doing in order to capture the big market, the public hospitals, insurance companies, etc., etc. The market, as you can imagine, cancer diagnostics is huge. 132 billion in 2022, and inside it, the liquid biopsy market is still a bit small because it's super early, 4.7 billion, but growing at a constant pace of 15%, expecting to reach 27 billion in 2035. Only assuming oncology, which is everything we're talking about today, but this liquid biopsy, and especially one with RNA, would be applicable to Alzheimer's diseases, coronary artery disease, and other indications. Looking at our competitors, as I said in the beginning, liquid biopsy is not something that we invented. It's something that people are trying to do and makes a lot of sense. Blood sample to detect signs of early cancer. We are not looking here at any other thing than cancer early detection. So everything that you're seeing here is cancer early detection in liquid biopsy. And you can do it by looking at different molecules. Most of the companies, as I said, are looking at DNA because it was a huge discovery when the first person described that it was possible to detect cancer DNA in blood because DNA is much more stable than RNA and other molecules. It looked right, and people went this way. The problem is that DNA is a very static molecule, and each cell only has one copy of mutated DNA. So if the cancer is still too small, which happens in the earliest cases, in the earliest stages of cancer, at that time there are very few molecules in circulation, and it's almost impossible to find it. It's worse than detecting a needle in a haystack because there are really few molecules to be detected. And on the other hand, RNAs are completely different because each cell produces thousands of RNAs. So even if the tumor is super small, you can have thousands of molecules that indicate that it has cancer, and even the cells around the immune system and all the tumor microsystem can produce RNA molecules that are indicative of the presence of the tumor. And we are capturing the sign not only from the tumor cells, but everything that is around it. And when we look at RNA, there are normally two different types of molecules. One is microRNAs, the other is just messenger long RNA or just RNA, which is the long RNA. So microRNAs are a bit more stable. There are some companies that are very advanced in the usage of microRNA for cancer early detection. The thing is that there are only 1,000 microRNAs in our body, different microRNAs. As I said, there are over 100,000 different long RNAs. So it's a question of scale. It's much more likely to have implication, or it's much more likely to find a positive molecule that indicates the presence of cancer if you have already 100,000 as a potential biomarker pool. And also because microRNAs are a molecule that regulates. They don't generate proteins. They don't do a direct effect except regulation. While RNAs are the ones that are making effects. They are the ones that generate proteins. They are the ones that activate the pathways associated with cancer, immune system, etc., etc. So you are monitoring more directly what's happening in the cells. And looking at the companies that are looking at RNA, there are very different approaches. We are looking at all the long RNAs. ExaiBio is a company from the US. They are using only orphan RNAs, which are very specific, obscure type of RNAs Honestly, I don't know why they decided to use this type of RNAs, but I think they are just looking at a small percentage of what's there. And Xinjiang is another company that is from China that is also doing research in the same field. They don't have anything published, we cannot compare to, but definitely in our vision, this is the future of cancer early detection, and many more people, many more companies will start looking at it. If we focus on the relevant deals here on the right, there were already some very interesting acquisitions in the past years. They are all in companies that were focused in the study of RNA, like Illumina acquired Grail for 8 billion, Exact Sciences acquired Thrive for 2 billion, which is crazy because it's much higher than the value of the market, and these companies never reach the market, not yet. And it's just because they know what's coming, and this shows the appetite of the big players for liquid biopsies. I will now let André explain our financial projections.
Yeah, thank you, João. So this is the classic slide where we make the projections for the future, no? Assuming all the costs we need for development and also the expected revenue in the coming years. No? This is assuming that 2026 we'll start already generating some revenue from research use only projects, and that we're going to enter the market in 2027 with some revenue, mainly focus on the initial indications of cancer, which will be colon, lung, and then continue with the pancreas, prostate, and breast. And then, these are projections, of course, and then we see this hockey stick type of graph that we all like to see. If we enter the market, we believe that it will go like this. If we are able to detect different types of cancer that early, it's really revolutionary when we expect something like this. So the main takeaway message from this spot is really, if we enter the market, it can be revolutionary and we can really go high in revenue. It might take more or less time, that these projections are always not easy to make, but we really foresee that the future is going to be liquid biopsy, hopefully with Flomics, and whoever takes this to the market should for sure will see something like this in terms of revenue. So there's a lot of details here, but the takeaway message is that one. Related to intellectual property, so we already submitted one patent related to colon cancer, and what we are protecting here are the biomarkers that we use to classify colon cancer patients. And this is basically a set of genes that we looking for patterns that allows us to separate the person that has colon cancer from a person that is healthy. Okay, and this was the first one. Now, once we finish the studies that we are performing also in lung, prostate, pancreas, and breast, we expect also to submit patents that will protect the biomarkers that we are using for these types of cancer. We haven't done it yet because we want more data in order to be sure about the genes that are included in these biomarkers. We are also about to submit a patent. These are like the final weeks, not related to cancer, related to a neurological disease that we work together with the Hospital Ronda that had really good results. And so it's not relevant for today, but just to highlight that we can also go for other diseases, not just cancer. And this, for example, is a neurological disease, a rare disease that affects young children. We also have trade secrets on the protocols that João mentioned. We are able to extract RNA with high quality, and we also protected the algorithms or the bioinformatics and AI methods that we have, and also protected the brand in Spain and in Europe. The following slide. So we are regulatory and certification, always a hot topic now. We are starting to work on this with different partners, mainly, I say here with Alida Health. We are working with them on the certification and regulatory roadmap. We actually already have the roadmap defined with them. Now we are working with them to define exactly what are the costs of the clinical trials that we're going to do for different types of cancer. So this is a tough topic, as you know, clinical trials and certification, and we want to be sure that we go with the right partner that can help us get certified and hit the market with the high quality standards. And I think I have one more. Yeah, the roadmap. So I'm sorry, it's a little bit dense. We try to put a lot of information. This is easier to digest, in front of you with time. But basically, where you see today, 2024, actually we're already in June, so more in the middle. So we are focusing on these preclinical trials where we want to have the final validation of actually 5 types of cancer. I want to highlight this. It's quite extraordinary. We have 1,300 samples from the hospitals that João mentioned. And then once we have these final results, this is going to be a very strong base to start the clinical trials that we plan for next year. And this is the first line you see. The second line more related to certification, all the ISOs, all the certification steps we need to do. And the second line, R&D. So we continue to do R&D to explore new types of cancer, new diseases, new opportunities. So we are very active in R&D. And then the following one, so we are writing patents, we are hiring people, we are already 18, João, I'm going to talk about it in the next slide. We also already generating some revenue because we do collaborate with hospitals and we provide services to others that will get us commercial experience, will keep us in touch with universities, research centers, and will also bring some revenue that helps us funnel to development and R&D. And then the funding part, so we are doing this round extension, as you know, kind of to complement the previous round that we did also with the Decelera Ventures. Lorenzo is present here and we'll talk about a little bit. And then we have planned for next year Series A of 15 million, still to have the final number because we're still learning how much the clinical trials are going to cost. But basically the Series A is going to cover the clinical trials and market entry. Okay, and that's the roadmap. I pass the ball to João and talk a bit about our.
Thank you. Just to wrap up, so we are based in Barcelona. We have our own fully equipped laboratory in Poblenou. If you want to come and visit us working, maybe do some pipetting, it's possible. Well, maybe not pipetting. I removed the invitation, sorry, but you can check it out. And we are a team of multidisciplinary scientists from bioinformatics, genomics, and also other complementary profiles. Lots of people with PhD with long track record and years of experience in RNA as a product like this requires. Also on advisory board, we have Rodrigo, which was my PhD advisor, a reference in RNA in the world, but also medical and business growth, everyone that we need to complement our skills. And at the moment, as you know, that's why you're here, we are opening a bridge round with Capital Cell. Now we have it in private, but we'll open it very soon, and June 17th. So depending on the day you are watching this webinar, if it's recorded, it might be already open. And this is 600,000 that we are aiming to. At the moment today, we are at 89% completion, so almost done. And this money will be used to complete this clinical validation of 1,300 samples and also to prepare the road for the Series A round of 15 million during the next year. We have to thank all our collaborators, clinical, but also institutional, and definitely also the sponsors, funding agencies from Catalonia, from Spain, and from Europe that made it possible. Thank you very much for trusting us. And with this, I finish. Thank you very much. And if there are some questions, I'll be very happy to answer them.
Pues muchas gracias, João. Yo creo que ahora te pediría que dejaras de compartir y le daremos el paso a Lorenzo. Así que, Lorenzo, te damos ahora el protagonismo para que te presentes tanto a ti como a Decelera y cuentes un poco qué has visto tú en Flomics para invertir.
Muchísimas gracias, João, André, por esta súper presentación. Y yo pido disculpas de antemano porque la mía va a ser mucho más breve y mucho más por encima. Pero antes de nada presentarme y presentar a Decelera. Decelera Ventures es un fondo de 40 millones basado con oficinas en Madrid y en Menorca. Y nuestra tesis de inversión es invertir en compañías del sur de Europa que utilicen la tecnología para tener un impacto positivo en el planeta. invertimos en fases de early stage y invertimos en Flomics en octubre del 2023. Y estamos invirtiendo también en esta ronda, estamos haciendo follow-on en la compañía. ¿Por qué invertimos inicialmente en Flomics y por qué vamos a invertir ahora de nuevo, ¿no? Entonces nosotros la forma que concebimos la inversión es principalmente en tres paquetes, ¿no? Uno, compañías que ataquen un problema muy, muy grande, con un mercado también muy grande y sobre todo equipos que sean excepcionales. Y de alguna manera explicaré nuestra decisión de invertir en Flomics basado en estas tres razones. La parte del problema creo que queda clarísimo con ese mensaje de 1 de cada 3 personas van a sufrirán un cáncer en algún momento de su vida, que este problema es enorme. Y nosotros, un pequeño caveat aquí, no somos un fondo especializado en health tech, somos agnósticos en cuanto a verticales, pero sí que nos rodeamos de gente de nuestro ecosistema que es experta en health y también en estas tecnologías. Y nosotros en nuestra decisión de invertir en Flomics sí que validamos la tecnología con players de la categoría de Pangea Oncology, Almiral, Universal Diagnosis y Topacium Oncology. Tuvimos esa validación y fue lo que nos ayudó, ¿no?, a decidir invertir en esta compañía. Luego, por otro lado, el mercado yo creo que está clarísimo, ya lo habéis mencionado vosotros. Nuestro análisis era incluso te diría un poquito más grande, es un mercado de 185 billion en la que se mejoraría muchísimo la calidad de vida de los pacientes y se reducen los costes que hay a la sanidad. Con lo cual, siempre me gusta decir que el riesgo-retorno es muy asimétrico, ¿no? Está muy enfocado en la parte del retorno. Eso, la parte del mercado y luego la parte del equipo. Entonces, aquí, de nuevo mencionar que nosotros en Decelera tenemos un modelo de negocio un poco diferente. Tenemos un programa de deceleración que llamamos nosotros en Menorca, donde todos los años invitamos a 25 founders. Nos aplican, este año ha sido en 2.300 y seleccionamos a las mejores 25 compañías y Flomics fue una de esas 25 en el programa del 2022. Y ponemos mucho hincapié en la parte de equipo, conocer a los fundadores detrás de las compañías. Los modelos de negocio ya están validados, es conocer durante 10 días a estos fundadores. Y la verdad es que, y luego desde el fondo de Decelera Ventures invertimos en las mejores compañías que pasen por nuestro programa. La due diligence de equipo de Flomics fue excepcional, fue fenomenal. Y tuviera que destacar algo del equipo que dirige la compañía, de João André, las 3 cualidades principales, pues diría que tienen un leadership skill muy alto, una pasión por el proyecto también enorme y tienen openness y transparencia, ¿vale? Son gente muy abierta y que están muy abiertos a recibir feedback y ayuda de la gente que tiene el cap table, y eso para nosotros es esencial. Por otro lado, tenemos la suerte, y ellos también tienen la suerte, de tener un cap table muy alineado en cómo vemos el futuro de la compañía. Tenemos inversores especializados como Pangea Oncology y luego también, como ha mencionado en sus slides, también tenemos un advisor de Decelera Ventures que es Genaro Bifulco, que también está muy hands-on en la compañía para llevarla a donde queremos llevarla. Y por último, simplemente mencionar que la razón por la que hacemos follow-on en esta compañía es porque vemos una proactividad muy alta. Al final, este es un mercado en el que hay que correr mucho y vemos que la atracción que está teniendo la compañía es muy buena a nivel de ese procesamiento de muestras. Nosotros, cuando entramos, la compañía tenía una especificidad del 70% y a día de hoy, como ha mostrado, está en el 87%, muy cerca de ese 90%, que es el benchmark que esperamos para esa serie A. No me gusta hablar solo, decir las bondades de la compañía, ¿no? Si tuviera que hablar de cuál es el principal challenge que nosotros vemos en la compañía, yo destacaría que hay que llegar a esa serie A con el procesado de muestras, de las 1.300 muestras, que eso es lo que va a validar las clinical trials, y tenemos que incrementar ese incrementado del throughput para pasar a un tamaño suficiente que nos permita llegar a final de año a estas muestras, ¿no? Ese es en lo que estamos trabajando y en lo que nosotros estamos haciendo hincapié dentro de la compañía para incrementar esa velocidad. Pero dicho esto, nuestra sensación con la compañía, con Flomics, es súper optimista y por esta razón hacemos este double down en la compañía.
Muchas
Y ya
gracias,
está.
Lorenzo. Muchas gracias. No, creo que has cubrido todo, entonces yo voy a ser más rápido porque no quiero repetirme, ¿eh? Creo que lo has cubrido todo y tenía aquí 4 apuntes de todo y si no, también hay alguna pregunta. Así que yo voy a decir solo de Flomics, un poco como Capital Cell, que estamos acostumbrados mucho a ver proyectos y estamos ahora acostumbrados a ver cómo evoluciona el mercado de la salud y nos estamos acercando a un mercado más, bueno, está a punto de colapsar el mercado de la salud, está claro, cada vez hay terapias más caras, los hospitales pagan más dinero y la prevención va a ser algo necesario. Entonces, todas esas herramientas que te detecten antes para evitar, lógicamente, reducir peores pronósticos, pero también para reducir costes, van a ser muy importantes y Flomics va por allí. Entonces, esto lo tiene muy ganado. También una cosa muy importante, yo creo que de Flomics, es que al final usa 3 tecnologías que se han juntado en un momento muy bueno, que a lo mejor hace unos años no estaban aún preparadas, que una es el ARN, el RNA, el otro es el Machine Learning, la inteligencia artificial, como lo queráis llamar, y después está el Next Generation Sequencing, que también es otra de las cosas que nosotros presentamos mucho como ejemplo de cómo estas tecnologías cada vez reducen más precio. Al final, Flomics está ofreciendo un producto con estas 3 cosas. El ARN ya lo sabemos, ha crecido exponencialmente a través de la pandemia y las vacunas. En el caso de la inteligencia artificial no diré nada, ya sabemos todo lo que ahora mismo lo tenemos todo y todo el mundo habla de ello. Y de Machine Learning. En el caso de Flomics es de verdad, ¿no? Porque a veces hay empresas que lo utilizan mucho, pero aquí hay que ver que se usa de verdad. Y una muy interesante también es la secuenciación masiva, que la reducción de costes que ha tenido durante muchos años. Esto no sé si lo recordáis y nosotros lo hacemos en alguna presentación. Hubo un proyecto para secuenciar un genoma humano hace unos años que necesitó toda la humanidad, casi, no, necesitó que mucha gente diera muestras y costó billions de 1.000 millones de euros para poderse hacer. Ahora mismo no sé los precios, pero se ha reducido mucho. Y básicamente la herramienta de Flomics de screening necesita ser una herramienta de diagnóstico que se pueda usar rápida, fácilmente, con una muestra de sangre y que sea barata. Y esto es lo que le permite estas 3 tecnologías. Y hablando también del éxito, que aquí yo creo que quiero reforzar un poco lo que ha dicho antes Joao, hemos visto éxitos comparables y aquí tenemos a Grail y Illumina que han comprado, han sido adquiridas o han adquirido a compañías por no millones, ni por cientos, sino por miles de millones. Y esto también hace un mercado muy interesante de lo que es los datos y el RNA y DNA. Yo creo que con esto no diré mucho más porque todo lo otro ya lo hemos dicho. Sí que es verdad que herramientas de screening son necesarias y aquí voy a apelar un poco a la gente, lógicamente, para detectarte antes el cáncer, pero también creo que es para evitar que te hagan pruebas invasivas. Para los hombres en el tipo de cáncer colorectal, pues nadie quiere ir a hacerse una colonoscopia porque sí. Entonces, con herramientas de Flomics ahorra esto y costes. Y igualmente en el caso de las mujeres, pues en cáncer de mama, pues la mamografía tampoco es muy cómoda, se utilizan rayos X. Entonces, estas herramientas te diagnostican antes, pero también ahorran costes a la sanidad y ayudan a los pacientes a no tener pues, inconvenientes ni preocupaciones.